Okay, so let's examine the machinery behind how that semi-conservative model of DNA replication actually works. Okay, we're now stepping beyond Watson and Crick into other people's work examining not only the enzymes but any DNA structures, sequences that may be of importance. Thing to keep in mind is that DNA replication is something that happens at extremely high speed. Okay, hundreds of nucleotides per minute. Okay, and it's something that has to occur with extremely high accuracy. Remember, in the last lecture I kept mentioning fidelity. Okay, you can't have a mistake where in the new strand where it should have been a T, an A gets laid down, or a G or a C. Okay, those are mutations that can lead to problems within the cell or problems that could be passed on to the next generation. What we do know is that replication is a very dynamic process. There are many enzymes, many proteins involved in DNA replication. I'm not going to hold you to knowing all of them or knowing the roles of all of them. That'll be something for senior level genetics. Okay, here for this course, we're just going to introduce some of the players. Now, one of the things you need to have is a place to start. Okay, there are going to be certain nucleotide sequences that are more advantageous for the beginning of replication. Keep in mind, the DNA double helix, the DNA is wrapped around each other. There are billions upon billions of hydrogen bonds holding this together. Now, individually, hydrogen bond, easy to make, easy to break, but you start adding billions, that's billions with a B, of those bonds together, well, now you have something that, it's a little harder to pull apart. It just doesn't fall apart. So you have to have a place where you can start this pulling apart so that the different enzymes and proteins that make up the replication machinery can get involved, interact with the DNA strands. This site is going to be a site, this region of DNA is going to be extremely AT rich. Why AT rich? Well, think about it. The adenine, the thymine have 2 hydrogen bonds holding them together, whereas guanine and cytosine have 3. It's easier to pull apart 2 than it is 3. So you're going to have this origin of replication, a place to start replicating DNA that is AT rich. For us eukaryotes, since we have linear chromosomes and we're going to have multiple chromosomes per nucleus, we're going to have multiple origins of replication. How many multiples? Hundreds or thousands, depending upon the length, the size of the chromosome. Whereas for prokaryotes, bacteria have one small circular chromosome. So they will only need one single origin of replication. So that is why you're going to see, if you ever read up the primary literature or go back and read on the experiments and the stuff we do to study how the machinery works, for the most part, we're going to always fall back to prokaryote models because one chromosome, one origin of replication, and it's just much more simplistic. Why start with a model that is going to be highly, it's going to have too many variables, highly complex, too complex for us to deduce all the components. So what we have been able to find when we study DNA replication in E. coli, what we have found is that there is one single point that everything starts, and as that is pulled apart, as that double helix is pulled apart, new DNA is laid down. You'll notice that this, what they call a replication fork, it's literally a Y. They call it a replication fork, this Y, because it harkens back to roads. Okay, you have a single road coming up, and then that road is going to split. That split, either go left or you go right. Okay, just like a fork in the road. Okay, the replication fork, the single double helix, is now split into two. And notice you have two replication forks. Two replication forks that are both working around the circle. This replication fork moving this way, this replication fork moving that way. They call it a replication bubble because as the forks spread, they make this new circle that's getting bigger and bigger, just like a soap bubble would as you blow more and more air into it. What's going to happen is the replication forks are going to work their way around until they meet on the opposite side of the chromosome. Now, this is different than us eukaryotes. Since our chromosomes are linear, you're going to have multiple origins, another origin. Still going to be pulled, the double helix still going to be pulled apart. You're still going to have a replication forks on either side. And these replication forks are going to go away from each other as the bubble gets bigger and bigger. Multiple bubbles, 1, 2, 3 here, as the artist has drawn. Now, keep in mind, if this was true, drawn to scale, that chromosome would have thousands of these all at once. As those bubbles get bigger and bigger, the bubbles will fuse, just like soap bubbles would. You blow and blow, 2 fuse to form 1, 5 form, 5 more will fuse together to form one bigger bubble, and so on. So you start at one point, and the replication forks work away from that origin. The replication bubble gets bigger and bigger until the replication bubble in the eukaryotes fuse together. One bubble, individual bubbles fuse together to form one giant bubble that all at once now becomes 2 distinct daughter DNA molecules.